Well, the first part of this lecture series on chromatin and DNA condensation, we introduced the idea of what does DNA really look like in a eukaryotic cell, and what is chromatin. Now what I want to do is to go back from that and talk about DNA condensation. How do we go from that G1 phase and what DNA looks like in sort of a spread out form at that point to these highly compressed chromosomes that you think about as classic dyads or metaphase chromosomes. So just to remind you, we talked about these things called nucleosomes. We said that they were made up of an octamer of histones plus this H1 histone that's found near the bottom or at an edge, and that the outer surfaces of these histones were mostly positively charged, which interacts with the negative charge of the DNA, which allows you to have electrostatic bonding. And that if you took the distance here from a linker DNA plus the couple of almost complete wraps, you have about 200 base pairs making up one nucleosome as far as the amount of DNA that's captured. And from top to bottom, we have about 11 nanometers. So we call this 11 nanometer chromatin fiber, or often called, again, beads on a string because of the way they look. So it looks something like this. If we are in G1, we have our beads on the string. These little dots again are nucleosomes. Most of the DNA at this level is either 2 nanometers, which would be just these naked coil of DNA. So the double-stranded DNA is about 2 nanometers across. The nucleosomes themselves, those areas are about 11 nanometers. So much of our DNA looks like this. Much of our chromosomes, I keep saying that because people only think this is a chromosome, but that's a chromosome right there too. So maybe, and it spends most of its life in the chromatin type configuration looking like this, not like this. Some of it's up to 30 nanometers. We'll talk about what that means in just a moment. And there's even some other slightly more compacted areas, but 30 nanometers is probably the more common type, right? So we have our DNA, we have our homologs, we go through replication, and by the end of replication, we produce sister chromatids. And as soon as we start to go through that replication process and we make that new DNA, those new sister strands are coming off, they start to compact. Nucleosomes form back quickly, and then those nucleosomes start to form together themselves into structures we'll call solenoids and other things, resulting in even a more dense packed structure known as a 30 nanometer fiber. So before we go into G2 and talk about further compaction, let's go take a look at what this 30 nanometer means. Okay, what it means is we've taken these nucleosomes and we have brought them together. One model shows it as a spherical cycle. Okay, and we've sort of wrapped them like this. Okay, so we've got our nucleosomes on our DNA, and we've wrapped it in a form like this, where we're forming a spiral as well, or sort of a helical type form to it. The distance across it now is about 30 nanometers. So we're pulling things down, pulling the DNA tighter, but we're having to expand the width, right? Because that's what happens when you pull something in a little bit tighter. Now, specifically what's happening here is that we've got this H1 histone, the special histone. It's binding up to these parts of the nucleosome here, this octamer, and the DNA, and it's binding up and pulling that very tight. It's also then got a traction between these areas of the actual H1 histones themselves. So start to pull those histones together in various ways. Okay, we know that from experimental data because here's this image of just our beads on a string. When H1 histone is added to those cells, to that mixture, that nuclear area, you can see that you start seeing compaction of these. You start to see this nucleosome pull close to that one, pull close to that one, in a fairly regular pattern. And if we were to measure from here to here, we would see that we've got about 30 nanometers on average across those structures, okay? And that's caused by adding this H1 histone. So we know that that's, as far as a research model, we know that that's one of the ways that it works, okay? Now, there are a couple of models about how it actually looks, and we know the distance, right? We know that across here you've got about 30 nanometers. One of the ways it's thought that it works involves this H1 histone pulling together between 5 and 6 of these nucleosomes as they make a circle. Okay, this is called a solenoid structure. It's a helical coil of these nucleosomes together being held in the middle by this H1 histone. Okay, pretty good experimental evidence that a great deal of our DNA, when it goes to compaction, certainly by the time we finish replication, actually looks like this type of form. It has this solenoid level to it. Okay, so we've pulled this together. Now, there's an alternate form known as the zigzag model, which in the DNA itself is in the same form as far as being around a nucleosome, but the nucleosomes are arranged in sort of pairs, and the DNA actually is a little bit straighter. So you don't have this open center that you have here, but in fact you have the DNA crisscrossing across in what's known as a zigzag pattern. And again, there's both experimental and observational data to support this. This is an image from a 30 nanometer fiber. Again, it's about 30 nanometers across, so it's sort of the same size. But you can see that the DNA, that the nucleosomes are not arranged in some nice circular pattern. They seem to be oscillating across in some zigzag pattern, as it's called. So there's pretty good evidence that both of these exist. The theory is that trying to take this DNA and bend it up enough to get over to the next nucleosome to make these nice perfect systems is the DNA simply may not be flexible enough to do that. The zigzag pattern allows it to be a straighter. You can kind of see this one right here. That piece of DNA would be a straighter connection over to the side. Then you fold around and you make a relatively straight connection back. So there's some idea that the linker DNA is less torqued by this. Okay, but when we really look, the experimental data says that both of these topologies really occur. They're apparently there, and they're in maybe even on the same piece of chromosome, same chromatin fiber. You may see both that are existing. One idea is that particularly the zigzag pattern is potentially associated with what's known as a chromatin hub. A chromatin hub is an area where you're trying to open the DNA up to read it, to read the actual code, and that this zigzag pattern is actually part of that folding or complexity that it goes through to kind of get these nucleosomes broken apart, are in a configuration where you can actually do the transcriptional process. We'll talk more about that later, but it's one idea that's where the zigzags may be. Okay, so and again, there's not a lot of good direct data on which of those are most common and why they're both there, but they certainly have both been observed. All right, so what have we done? We've taken our DNA, we've coiled it, we've wrapped it around these nucleosomes, we've pulled it together into some type of configuration. A lot of it is solenoid, pretty clearly. So we're right in here, okay? Well, that's certainly some of that's in G1. There is a bit of this coiling. Most of it's like this in G1. By the end of replication, almost all of our DNA is at least in this form, this 30 nanometer form, okay? But then how do we get from there to these compacted up metaphase chromosomes that we think about? How do we make it to those, okay? Well, one of the models involves what is known as higher order coiling, which has several steps. The first step is to sort of make a 300 nanometer fiber, and that 300 nanometer fiber is produced by what are known as chromatin loops. These are chromatin loops that you see here. So these are solenoids that then are looped out into some configuration like this. And the basic idea is that you have the nucleosomes. This is all solenoid right here, solenoid wrapped around, folding physically, sort of solenoid, and it's physically folding out like some form like this, attached then to some central area. These are generally known as loops, and then when you get several of these loops formed around a central core, that's known as a rosette. So we have loops, we have coils, we have rosettes. All of these are looping and containing areas. So these are the loops right here. And then you start to have a more condensed form where you start to see coils and other things, okay? Now, in order for all this to work, you've got to have something to attach to. You've got to have a central core here, right? You've got to have all of these loops just aren't going to just form out there and sit there. They've got to somehow attach to things. So what we think is, and what's been proposed at least, is that there is a series of specialized proteins that help form a scaffold. You'll talk a lot about scaffold when you take cell biology and other things. in the cells themselves, proteins form these structures, right? They form this part of the actual mechanical skeleton, if you will, of a cell. And these are called scaffolds. And so the scaffolding here, so it allows these loops to connect and form these basic structures. So we see these loops, we see this scaffold form, and we start to see more coiling. So we've got loops. The loops form rosettes, so they're around like this, and then those rosettes pack together in even more and more dense form, so that eventually we end up with the DNA packed up around these coils, around these rosettes, into these very complex areas, okay? And that's what gives us that fuzzy appearance. Now, how do we know this stuff is even there? Well, experiments have done where you can isolate the DNA, and once you remove the DNA, what you end up with is this structure that looks something like this. Okay? And what we're seeing here is the actual protein scaffold that comes into place. So if you actually look at this, this would be one leg of one of the dyads, one of the sister chromatids coming along like this. And this would be some region that would be the actual centromere that's found up in here. So this would be one dyad, part of the dyad over here, one sister. This would be the other side over here. And all we've done is remove the DNA. So the fuzzy part that you saw in those first pictures of DNA are no longer there. Okay. So we know that there's a scaffold of some kind. We know something about the forms of those scaffold. All right. So what have we done? Overall, we've taken double-stranded DNA by coiling it. We've reduced the length there. We've then added nucleosomes to it. Okay. And then once those nucleosomes form together, they complex up into a structure that's either in a zigzag or a solenoid type pattern. They further coil and rosette and pack around until finally we end up at the end of G1, somewhere up in, this, or excuse me, at the end of G2 in an early stage of trying to make this final chromosome. Okay. Now, we'll walk through that in a moment, but what I want to point out is we know quite a bit about the nucleosomes. We understand them. You can isolate them. There's kits you can buy to get nucleosomes out. We know that the tails are activated and deactivated by acetylation, as well as other things that methylation and other things can occur that cause reactions to them. We certainly know that these chromosomes exist like this, these dyads in these very dense forms. We know that there's some fairly regular things that happen in here because we can measure and see that there's pretty much a certain size that everything is in at some one point, and then it becomes a more dense size at another point, and eventually we end up here. The reason I put this little red thing here is that's my black box. We still don't know exactly what's happening. It's one of those nice areas in science where we're still working on it. We know that it happens. We know that we end up with these forms, but mechanically, exactly what's happening, which proteins are becoming active, what enzymatic activity is going on to help this folding, it doesn't just do it by itself, is really still an area of intense investigation and new ideas and new data coming out pretty consistently. So I'm going to call that my little black box. Okay, I'm going to tell you that it definitely happens. We have a pretty good idea of the order and pattern there, but we don't really know the mechanism, which is kind of fun. And that's much of science. We don't know a lot of the mechanisms. All right, so just to take you through this, remember that G1, we're about at 2 nanometer fibers, naked DNA, up to 11. This will be 11 nanometer fiber here. By the end of replication, we're pretty much everything's in a 30 nanometer fiber. Early on in G2, we're about 300, so we're starting to form those little things we call rosettes based on loops. And we take those rosettes and we start to coil them around this central area that we will have some type of scaffold made by protein. And then finally, everybody's memorized PMAT, prophase, metaphase, anaphase, telophase. And it says one of the things happens in prophase is that it's the final shortening and thickening of the chromosomal material. And that's exactly what happens. We get this scaffold, we get this final compression of our chromosome down to hundreds of times smaller than what the actual chromosome is over here in G1. Right? So let's just summarize this for these two lectures. First, we got these big old pieces of DNA. We got 2 meters of DNA in every cell, at least in mammalian cells, and similar amounts in others, plus or minus. Some things have more, some have less, but they've got to pack down into a pretty much standard cell nuclei So they've got to be packed down, just normally packed down, just condensated, just to fit, period. There has to be some level. And then the this condensation has multiple steps, right? We pretty much believe there's a nucleosome model, good evidence for that, that there are these nucleosomes. They have this special regular structure. We understand that there's coiling of those nucleosomes into either solenoids or zigzags or something, but pretty much a 30 nanometer fiber that we see. And then once we really need to coil it, once we really start the process of getting ready for mitosis or meiosis, we go into intense supercoiling. This involves loops, rosettes, coils, all the structure. And eventually, once we get really close to prophase, so we're really late in G2 and we're about to enter prophase, we see the appearance of these scaffolding proteins. And those scaffolding proteins are what finally help all this hold together. And they are very active during the period of prophase and into early metaphase. Okay? And so we have this compaction down. Once we go through metaphase and anaphase, start pulling those things apart, that DNA actually starts to spread out again, because by the time we get through telophase and we get that cell replicated and pulled apart, we want the DNA to be available again. So it folds back into these 11 nanometer beads on a string family. So we go through this process. Okay? Again, most of the time that we look for DNA, most of the time we look for chromosomes, they're actually in this beads on a string modality. Okay, that covers pretty much supercoiling. Again, there's lots of more information about it if you want to look on the web or other places that will tell you about it. But this will get you through the idea. And I'm just trying to build a picture of, like, this is what chromosomes look like most of the time, which is DNA with nucleosomes. And this is what they look like for a little bit of period of time when they're in meiosis or mitosis, and we think of them as these original chromosome structures.